Okay, good afternoon, everybody. So I'm Jane Heffernan, and I come from York University and also the Center for Disease Modeling. And for the next lecture, we're going to talk about why make models. And I thought that it was important to understand first where we see math in biology. And so we're not going to jump right into disease modeling. We're going to look first at math in biology and look for some explanations of how we see some things in nature. Okay. One second. Okay. So this is a Fibonacci sequence. I'm sure you've all seen this sequence of numbers before.
Yeah.
Okay. So when I go to elementary schools or high schools, I always start out by showing the students this Fibonacci sequence of numbers because it's a basic model, I guess, of things that we see in nature and also in architecture. Okay. So we have 1, 1, 2, 3, 5, 8, and so on. And what I show to the students, like I bring in some flowers, and we see that the number of petals of a flower is a Fibonacci number. And if you see 6 petals on a flower, usually it's 2 rows of 3 petals. And so I have students go out into nature, and I bring things into class too, to look for the Fibonacci numbers in the world around us. Okay, but then related to the Fibonacci sequence is the golden ratio. And so when we look at this golden ratio and you draw your squares of the Fibonacci sequence and you keep them this way, you get this nice spiral. And that spiral can also be explained, it's a golden spiral, by the Fibonacci numbers and the golden ratio. And you also see that everywhere in nature. And so usually, so if you're going to go visit schools, you would bring some things with you, like a pine cone, so that they can see it. Or if you go to the grocery store, maybe you can show them the pineapple, which gives them the spiral in 3 different ways, but also the number of spots in the pineapple, the number of sections in the spiral is also a Fibonacci number. And so we go through and we do some counting. Okay. Also looking at nature, we can look at the stripes and spots that we have on animals. You can naturally recognize, if you owned one of these cats, you would recognize which one is yours because of the patterns. And there's some really cool modeling that you can put behind how these patterns form. And the same model can be used to give us stripes or spots or other types of configurations on animals. And so we can use the same model to give us these patterns. And all we're going to do is just change some small parameters in the model. And so if you're interested in looking at stripe and spot formation, you can look at a textbook by Jim Murray called Mathematical Biology. Okay. And so we use, or we can see in nature things that we also consider to be math. So stripes and spots are mathematics. Okay. I just want to also say that when we look at what we have in nature, we also see fractals everywhere. So you can think that a branch on a tree is a small tree, and a twig on that branch is also a small tree. A small grain of sand is part of a rock, which is part of a larger rock, which is part of a mountain, right? Those are all fractals. Okay. And so we can also consider the mathematics of fractals behind everything in nature as well. And fractals are models. Okay, so here is the Mandelbrot set, which we do see in, say, movies. Like some of these things that we have on the edges are used to like make lightning bolts in movies. And this spiral that you see in the Julia set, which is related to the Mandelbrot set, is also something that we see related to spirals that we have in other things. Does anybody know what this is? It's a Brocco flower. So it's the cross between a broccoli and a cauliflower. And when I show students this fractal, the Julia set, we then also look at a Brocco flower and we can see how they're related. And then also looking at this, you can see the fractals. You see a large Brocco flower, and each smaller section of the Brocco flower just looks like a larger Brocco flower. And you see the spiral going up it too, and that's the Fibonacci sequence, or Fibonacci numbers, or spiral. Okay. And so when taking into account all of this math and biology, when we're watching movies, we actually see a lot of computer-generated mathematical models in the movies that we're watching. And so this is not actually a picture of a hurricane from space. This is a computer-generated hurricane cloud using fractals and spiral models, right? This mountain looks real, it's not. This is a computer-generated mountain using a recipe of a fractal. Okay. And so when we're looking around the world, or in the world around us, we're looking at all these things, and we can always interpret these objects that we're seeing in terms of mathematics. Okay. And so when I saw the question, why make models? My answer was, well, I want to understand the world around me, and the models help me do that in a quantitative way, as well as in a qualitative way. If any of the other instructors or any of you had been given that question, you probably have a different answer. But this is what I came up with. So I make models to understand things in biology, chemistry, physics, social science, psychology, health, etc. Whatever I'm interested in. I model immunology, I model behavior, I model honeybee stress, and for pollination jobs. All of these things I want to understand essentially the pattern in the world around me. And so I use models to look at things in a quantitative way, but also this is something that people outside of mathematics and mathematical modeling don't quite understand, is that math doesn't always need to be quantitative. It can be also looking at things in a qualitative manner. How do we get oscillations? I don't care about how big the oscillations are, and if the magnitude on the y-axis on the graph is telling me a quantity. I just want to look at the qualitative component as, do I get a big oscillation and then a small one? If I add stochasticity onto that, maybe when I add in noise, how much can I get rid of the oscillations, or do they get bigger? So I just want to look at the qualitative component as well. And so when I was a grad student, I was really interested in trying to understand, and a postdoc, trying to understand. So as a grad student, first, I was interested in understanding this outcome that comes from HIV patients, where they will have a viral load that's undetectable. And then once in a while, they'll have a detectable viral load. So it's called a viral blip. And so can we develop mathematical models that will give us undetectable viral load in individuals, where we can add some stochasticity or some other biological mechanism that will give us that blip that doesn't mean that their viral state is actually changing. It's just something that just happens naturally. So how can we model that? As a postdoc, I became more interested in trying to understand childhood disease incidence in populations. And so we look at these periodic outbreaks of different diseases in the population. So this is measles in the UK, varicella in Ontario, and rubella in Ontario. And you can see that these diseases have this periodicity to them. And so we're going to talk about later in this course about how to induce periodicity using mathematical modeling. But we also want to, so that's like using that to that type of model can help us understand periodicity in outbreaks. But we also want to try to understand what's making these periods. Why are we getting outbreaks in this like biennial manner for measles? And so I'm interested in mostly in immunity. And so I'm interested in trying to understand how immunity is changing in a population. So this pattern of immunity, such that we also get this pattern of oscillations in the population. And how that changes depending on what population we're looking at and what pathogen we're looking at. Okay. And then related to disease spread, we're also interested in understanding the patterns of spatial spread of infectious diseases. And so here is a graph of how foot and mouth disease was transmitted in the UK. What year was that? Does anybody remember? When was the foot and mouth outbreak? Was it 2000, early 2000s, right?
It was 98, and then it was 2001.
Okay. And so so we look at spatial spread, and you can think of, if you've done some spatial spread modeling before, you can think about like dispersal or by wind or things like that, some dispersal of the pathogen in the environment, but also in terms of trying to understand this weird pattern of spatial spread of foot and mouth disease in the UK, people then also had to understand the movement of farmers and how farmers drove around and talked to other farmers, maybe sometimes far distances. And they also needed to understand the geographic location of markets, where farmers take their livestock to market, and how the livestock intermingle with other livestock in these markets in the UK, which can aid in disease transmission. And so you can get a pattern over time where you'll see like different fluctuations of cases and incidents over the UK. But these movements of farmers and animals in natural trade in the agricultural sector, those patterns are also important for us to try to understand, to tease out of data so that we can make a better model. And so I was talking to some of the other instructors at lunch. I was trying, why do you make models? And there was a really good answer about trying to understand data. There was also the comment from the late Fred Brower, what do we need data for? We can model things that are observed in a qualitative manner. We don't always need data. But really, like, we just see these patterns around us. We see biology in the world around us, and we want to use that to inspire us to make models. How do we make models that give us these outcomes, these patterns, right? Okay. So what I want to do then is give you an introduction to some of the first models in mathematical epidemiology. Okay. So this is Daniel Bernoulli. How many Bernoullis were there? 3 or 4. Yeah. So this is one of the relatives. I think there was a father, an uncle, and 2 brothers. Anyway, so this is Daniel Bernoulli. And in the 1700s, there was work done by him to plot out the cases of smallpox in the Kingdom of Sweden. So this is an actual plot, something that he did, where he was looking at the outcome of vaccination, a vaccination program to combat smallpox. And he's showing how you have here, so here there's a graph of smallpox with measles, and this is a graph of cases of smallpox, and then so this is before vaccination, and then after vaccination, we get these lower outbreaks of smallpox. And so this data, or this accumulation of data, this first type of example of a model, was used to publish in like I think it was like a local newspaper, to get people to go get interventions so that they could limit spread of infectious diseases in a population. Okay. So here is another example of a very early study in mathematical epidemiology by Jon Snow, not the Jon Snow from Game of Thrones, different Jon Snow, where there was an outbreak of cholera in London, England. And it was disturbing to the locals to understand how cholera was being spread in their small geographic neighborhood. And so what he did was look at a spatial model. So plotted out where all of the cases were. So these are bars. So these are addresses, and the length of the bar is the number of cases in those households. Okay. And he used this information, then used a simple center of mass calculation to figure out where the center of all these cases were, and found that it was close to a water source on Broad Street, called the Broad Street Pump. And so he used this simple model to lobby the government to stop people from using water from that water source. And lo and behold, when that water source was limited or capped, the cholera outbreaks decreased and it went away in that location. So then that helped instigate a study into why that pump was causing cholera. And the epidemiology study showed that the intake of water for that pump was directly from the Thames River, of course. And just like a little bit further up on the Thames River was a sewage release into the river. And so people were drinking or using water that was contaminated with sewage, and that gave them cholera. Okay. So use statistics to show connection between the quality of the source of water and cholera cases. Okay, so this is an example that you may not have heard of before. This is Ignatz Philippe Semmelweis. Has anybody heard of him before? Okay. Well, of course, Amy has. And he is known as the savior of mothers. And so what happened with him, so he was the director of this hospital in Vienna. So he's a Hungarian physician. He was the director of this hospital in Vienna, if I remember correctly. And they had 2 obstetric wards in the hospital. One was run by midwives and one was run by doctors. And this is in the 1800s. So who were the midwives and who were the doctors? Men were the doctors and women were midwives. Okay. Now, in the 2 obstetric clinics, there's this disease called puerperal fever. And this disease can be very drastic in outcome for the baby. I think it's just the baby. Is that also the mother? I can't remember.
It's bad for the baby, but the mothers can also develop like systemic infection.
Okay.
Bad for them.
Yeah. And so you can see that one clinic had a higher yearly mortality rate than the other one. And the first clinic that has the higher mortality rate is the one run by the doctors. The blue one is run by the midwives. So that was disturbing to Semmelweis as well as the community, because at the time, it was believed that doctors were smarter than midwives and men were smarter than women. And so how is this going to happen where we have a clinic run by men that has a worse outcome than a clinic run by women? What was also very disturbing to Semmelweis was that the community around the hospital knew that this was the case, and these clinics were open on different days. And so if you as a pregnant woman went into labor on a day where the first clinic was open, you didn't want to make it to the hospital in time. But in order for you to qualify for public health care, you had to show that you were on your way to the hospital. And so people were choosing to have their baby on the street. They couldn't have their baby at home because then they wouldn't get public health care. They were choosing to have their baby on the street on the act of going to the hospital, and that being safer than actually getting to the hospital and having their baby in the first clinic. Okay. And so Semmelweis, what he did was an epidemiological study, but inspired by data. And so what he did was go through the pattern of what the midwives and the doctors were doing during their day. And he found that the doctors, before they were doing rounds, and before they had their rounds in the obstetric ward, they were actually in the morgue learning about internal anatomy. And this is in the early 1800s. And so this is before the germ theory of disease. So they actually had their hands in dead bodies, like in bodies full of bacteria. And then they would just wipe with a towel, wipe their hands off, and then they would go to the obstetric ward and they would deliver babies. And so he instigated hand washing into the hospital, and the incidence, the yearly mortality rate at the first clinic then reduced down to the same as the second clinic. But then, because no one believed that if you can't see it, it doesn't exist, right? And so behavior waned in the doctors, and the incidence or the mortality rate in the first clinic went up again. And then, so there was this public health battle, with the doctors for a while to get them to wash their hands. This actually drove Semmelweis crazy. He went mentally insane because people didn't believe him. If you don't see it, it doesn't exist. And he actually eventually killed himself because he just was driven crazy with this whole thing. Okay. So another example, this is Ronald Ross. Anybody heard of Ronald Ross before? He won a Nobel Prize for his studies on malaria. And he developed mathematical models of mosquitoes and malaria transmission between humans and mosquitoes. And his modeling and biology studies were so well received by the community and the scientific community that he won a Nobel Prize. And his work to this day is like the basic model of malaria. And he showed that control mechanisms of certain populations or certain mechanisms of control were better than others, using, it was like a 2-equation model. Okay. So I can't remember what textbook this is from. I should actually put the citation here. I was looking at a textbook on epidemiology, and I was surprised to see they actually had disease modeling in their Venn diagram I don't know how many Venn diagrams people use, but I see my kids, I think every week have to write a Venn diagram about something in their classes. Okay, so, but what they have this Venn diagram of epidemiology, and they have research, epizootic disease control, there's connection, and community-based animal health programs, so you can see the intersection. I don't know why there isn't a middle here. Okay, because you'd think that those three should, you know, have one common thing. But also the disease modeling is actually in the Venn diagram, but it's over here. Okay, and so disease modeling has nothing to do with community-based animal health programs, Amy.
I just like this guy.
Yeah. And also it's not shifted over to like have this intersection between research and epizootic disease control. So there are lots of things that are wrong with this Venn diagram, but I was very happy to see that they actually were thinking about disease modeling at all. But there's a star there, an asterisk. And I won't go into what that asterisk actually said. But anyway, so I can say that over my career, disease modeling has been around for a while, and we'll see on the next slide the basic model of disease modeling, which we've already talked about a little bit today. But over my career, disease modeling has actually changed quite a bit. Going from mostly I do immunology studies, looking at the multi-level studies of within the cell to the body, to between bodies, to populations, the feedback in between, looking at the connections between animals and humans and insects, all these things. And so I would say that this disease modeling circle should actually be bigger than this entire Venn diagram, and that the rest of the diagram should be inside it. Because mathematical modeling looks at the basic research. We use models to help with basic research in epidemiology of endemics and epizootics and studies of new diseases and reliability and validity of studies on all of these things. So yeah, disease modeling should be the big circle, and all this is inside. Okay. And so that brings us to that basic model of in mathematical epidemiology is the SIR model developed by Kermack and McKendrick, which give us the first building block of what we all are going to work on this week, and what we're learning, and what some of you are doing your research on for your graduate programs. And I don't know if you have actually read the papers by Kermack and McKendrick, but if you haven't, it'd be very interesting for you to go back and read them, because they're written in terms of a story, they're like a story. It's very much prose. It's not so much a lab report. We write papers as lab reports more or less now, but they're prose, and it's really interesting. Okay. So we develop, why make a model? So we make models, various different types of models, to try to understand patterns in the world around us, or data, or even to supplement data. If we don't have enough data, we can use models to help provide some data. We also use models to help supplement data sets for machine learning. So people are going into AI and machine learning in disease studies. Virtual clinical trials or in silico experiments are used to help educate machine learning, so that we can have better study data sets, as well as outcomes in terms of individualized health programs. Okay. And so you will be faced with why make a model. Has anybody told their family that they're here? And has your family asked you, well, what's that? Or what are you working on? How are you going to explain what you're doing? One thing that was interesting is looking at how the community and our families became more aware of models during the COVID-19 pandemic. There were models in the news. There was even a huge news story. It was that Neil Ferguson, a mathematical modeler in the UK, wasn't abiding by the public health mitigation strategies in the UK. And it was huge news that he had to, he gave up his position in the World Health Organization advising committee, because he wasn't actually living in social bubbles like he was supposed to. But modeling became more and more commonly known and in discussions between people in the community. And so before COVID, people would ask me, what do you do? I said, well, I am a mathematical modeler. And they're like, what's a model? Now people are asking me why, like, what am I modeling? Why I'm making the model for that? What am I trying to understand? What pattern in the biology? I play soccer, and on the bench, when I'm on the sidelines with some of my teammates, they're asking me what I'm working on now. And so there's lots of interest. There's all this modeling in the news, and people are also trying to understand why we make models. So why make a model? Understand patterns, look at data. Why are we making models? We try to help others understand why we're making models and what we're trying to do. Okay. So the questions that we get are, what's in a model? What do you do? What do you look for? What data do you use? So sometimes we say, I don't use data. I don't have to use data, especially if I want to look at qualitative outcomes. Okay, and some people don't like that answer. What mathematical tools do you use? I like to say calculus, which scares people. Are models right? If a model is wrong, what does that mean? Or I should clarify that models are wrong. What does that mean? And so all these questions, I will have different answers to, or we'll all have different variations of similar answers to as well. But these are things that we all have to think about. People are more knowledgeable about models than ever. And so you will be asked questions like this. Okay, so you can think about your learning goals for this course on why make models, what are we making models for? It's not just fun. I mean, I like that answer. I think it's fun. But, what are we actually doing? We're trying to publish papers. It has to be something that's, helps other people understand things. And even if it's just for bifurcation dynamics, you can make models because you want to do bifurcation analysis. That's an okay answer. Okay. So, but for me, so the basic, our learning goals are to understand the basic model structure underlying all models of infectious diseases. That's the SIR model. How is it inspired? How do we add complexity to those models? We also have the basic model structure underlying the in-host modeling, which I'll be talking about later this week. So the immunology. We want to gain understanding and inform public health and medicine. And so we want to understand this reproduction number. I can't believe how many people are still asking me about reproduction numbers. So the COVID is increasing right now. My family's asking me, what's the reproduction number? Herd immunity, social distancing, heterogeneity, all of these things, we can use models to gain understanding. And then as part of this week as well, or week and a half, you'll be reading and discussing modeling papers with an understanding and critical eye. My problem sets are going to not all be math. There will be writing reflection papers, discussion things on things that we're reading and talking about in class. But it also helps us interpret news stories involving infectious disease models. So there's a lot of spin on modeling in the news. There's lots of discussion about that. And so we also have to understand when we're reading these news stories, how they're reporting it, a lot of the reporting is incorrect. Some of the words, the terminology is incorrect, which we talked a little bit about with Amy this morning in terms of terminology. but also things can be made bigger than they actually are. And so it's important for us as modelers also to read these things with a critical eye, and to understand that when we're interviewed by the media, how what we say can be spun to the media as well. Okay. So this is my last slide, because I wanted us just to have a short discussion session with people around you on why you make models, what you're making models for. You may not have been able to introduce yourselves to everybody in the room as well. But I also wanted to just leave everybody with this figure before we talk about modeling. And this is something that I use in my group, where so a lot of us are going to work with biologists, immunologists, epidemiologists, people in public health. And there's this want usually for us to incorporate more and more and more of the biology into our models. Okay. We also have to consider what we know already about the biology and what data we have, if we do or don't have data. Okay. And what we want from our models is we want some new knowledge from our results. Okay. If our models don't incorporate enough of the biology, we're not going to get any new knowledge. Okay. If our models incorporate too much biology, we'll get results, but we won't get any new knowledge from our results, because we don't have enough data or understanding of the biology in order for us to be certain that our results are valid. Okay. And so if we're incorporating, the more biology we incorporate into our models, the more uncertainty we're also going to incorporate. And then that means that we have to do sensitivity and uncertainty analysis. And we can get a whole whack of crazy information from that. Okay. So we don't want to have not enough biology. We don't want to have too much biology in our models. We want to have just enough so that we get that optimal level of new knowledge from our results. And getting to that point can mean lots and lots and lots of iterations of making a model, revising the model, debugging your code, making the model again, discussing, going and talking about your work to collaborators or people in knowledge users, going to give presentations at conferences, taking all that information back and going back and revising the model over and over and over again. And so I've been in disease modeling for a long time, gray hair, and I'm still revising projects that I worked on as a graduate student, because now we know more and more and more. How can I extend on those projects so that I can get more and more new knowledge from my results with less and less uncertainty? Okay. And so we have about 20 minutes left, or wait. Yes, my watch is on the right time zone. Okay, so we have about 20 minutes, and I was thinking that people here, I don't know if you want to go through the room and introduce yourselves, or we can just talk to the people around us. Amy is not here, is she? What do you guys think we should do?
I would say maybe just form a little bit like
Yeah, turn around to the people around you and talk about what you make your models for, or are you learning how to make models and what you want to make a model for, what the patterns are that you're trying to understand, and so on. So I'll just stop there.